Good morning, everyone. My name is Hajime Onuki from Ajinomoto. Thank you very much for coming to today's seminar regarding on the consistent and efficient hPSCs cell culture system with a novel animal origin-free culture. And we are facing a very severe situation due to the new coronavirus, but I hope you enjoy today's seminar, and this seminar will help your stem cell research. So let's start it. So first, I'd like to introduce our company, Ajinomoto. So the headquarters is located in Tokyo, and we have 3,000 employees in the unit, and we have more than 34,000 employees as a Ajinomoto group. Actually, we are global leading amino acid suppliers, and we have long experience in providing a clinical grade amino acid. In addition to amino acid, we are also providing seasoning and processing food and frozen food and some healthcare product such as cosmetic and culture media for stem cell research. You know that the first human ear cells was established in 1998 by James Thomson, and first human iPS cells was reported in 2007 by Dr. Shinya Yamanaka and also James Thomson. And now there are many progress in the stem cell research field in both basic research and clinical research. This table briefly shows some examples of clinical research by using human iPS cells. The first human iPS cells clinical research was performed by Professor Masayo Takahashi in 2004, and she transplanted autologous iPS-derived retina cells to the patient. And she reported that there was no adverse event 1 year after transplantation, and this report suggests that the potential safety of human iPS cells. And now there are many types of clinical research or clinical trials by using human iPS cells. And in future, this number of clinical research are increasing year by year. Although there are many progress in the stem cell research, there are still some challenges of the pluripotent stem cell research. For example, inconsistency of experimental result and regulatory barriers for clinical application, and labor-intensive cloning process and expensive culture cost. So these are challenging for the stem cell research. Some scientific reports clearly show that the culture reagent, such as culture media, growth factors, or scaffold protein, affect the quality and safety of cell product. Especially the animal or human-derived reagent, such as a BSA or BSA-containing reagent, and FBS or mouse-derived scaffold, such as matrigel, has a potential risk of the inconsistency and unsafety. For example, such animal or human-derived product has the risk of the immunogenic contamination, and also such product has large variation through the lot to lot because of the difference of donor. In the regulatory aspect, the animal-derived component is recognized as a risk for cell therapy. US regulation, USP 1043, clearly describes that animal-derived materials has higher risk compared to the other product. So these animal-derived products are not ideal for the cell therapy or the manufacturing of cell therapy product. So that's why animal origin free is important. This slide shows the animal origin-free. What is that animal origin-free? So, for example, like culture media, there are some type of categories like serum-free media or xeno-free media or animal origin-free media. The serum-free media can contain the BSA, and xeno-free media sometimes contain the human-derived material like human serum-derived HSA or human platelet lysate. And this animal or human-derived product has the potential risk of the immunogenic contamination or lot-to-lot variation. On the other hand, animal origin-free product does not contain these human or animal-derived product, so it has negligible risk of the immunogenic contamination and minimum lot-to-lot variation because it does not have the donor variation. So that's why the AOF culture system supports the consistent and regulatory compliant cell production. So we recommend to switch from the animal or human derived product to the AOF product because such human or animal derived product has a risk of the variation and sometimes it is prohibited for the cell therapy product. So we are providing AOF culture media, StemFit, and we think that this media can be solution for stem cell research. This StemFit product has 4 big features: consistent performance and regulatory compliance, and easy single-cell cloning, and significant cost reduction. This slide shows the performance of StemFit media. These data are provided by a British company, GCT Catapult. They compared the commercially available culture media for the pluripotent cell expansion, and they found that the StemFit media showed the higher performance in the cell expansion, and also iPS cells cultured in StemFit media maintain the similar gene expression profile through many passages. So these data show that the StemFit media has the higher cell growth performance and also the stable gene expression profile. Also, StemFit media is widely used in the clinical research field. In Japan, StemFit media was officially confirmed by PMDA. This is the Japanese authority for the pharmaceutical and medical devices to be usable for clinical application. And in United States or other country, GMP compliant product is also available as necessary. Now, the clinical grade iPS cells or ES cells are manufactured by using StemFit media. And also, many clinical research for iPS-derived cell therapy are going on with our StemFit media. For example, world first clinical study with iPS-derived corneal cells is proceeding with StemFit media because StemFit media is regulatory compliant and it's usable for the clinical application. And currently, we launch the StemFit for differentiation supplement. This is a serum replacement supplement for the iPS ES cell differentiation. This supplement is also animal origin free and can be used for the differentiation. For example, this slide shows the performance of the endoderm differentiation, like a hepatocyte. Usually, a B27 supplement or KSR are used in the differentiation process, but these products sometimes contain the BSA or animal-derived product. So it's not ideal for the stable stable differentiation. So we replace these supplements to the StemFit for differentiation supplement, And we found that the mature hepatocytes was obtained with high quality and high gene expression performance. And this supplement can be used for other type of cells like mesoderm and also ectoderm. So this supplement can be used for 3 lineages, including mesoderm, endoderm, and ectoderm with high quality and high performance. So we believe that the animal origin-free product can be a solution for the inconsistency and regulatory barriers. But there are still some challenges, such as a labor-intensive cloning process and expensive culture cost. So with StemFit, the easy single-cell cloning is available because StemFit shows the higher cloning performance compared to other commercially available media. iPS cells or ES cells do not grow well under very low seeding density, and that's why the single-cell cloning is difficult for pluripotent stem cells. But stem cells cultured in StemFit media showed higher cloning efficiency compared to other culture media. So this media, StemFit media, enable a very easy and simple single-cell cloning. And also, StemFit media contributes to reduce the cost of the culture media because it enables weekend-free feeding. The conventional culture media required daily medium change, so researchers need to change the culture media every day. But StemFit media only required 3 times or 4 times medium change per week. So this feature contributes to reduce the cost of the culture media. For example, the conventional culture media requires 80 ml 80 ml volume per well per week because researchers need to change culture media every day. Or other weekend-free culture media required 30 ml per well per week. On the other hand, StemFit only required 7 ml per well per week because the StemFit media does not require double feeding before the weekend. And in the 6-well plate, this media only required 1.0 ml culture media. On the other hand, other commercially available media usually required 2.00 ml per well. That's why StemFit media enable the significant cost reduction compared to the other commercial media. So this slide shows our main AOF product, Basic 03, and currently we launch new product, StemFit Basic 04 complete type. This media is the optimized version of the Basic 03 and composed of only 1 bottle. So it's completely ready to use. This media also has the more affordable pricing. So, and as I explained before, we launched the StemFit for differentiation. This media enabled to manufacture the target tissue and the AOF culture condition. And Ajinomoto also providing growth factors. These growth factors are also animal origin free and enable to manufacture the target tissue and the AOF condition. So, as a summary, the first, animal or human-derived product has potential risk for inconsistency and unsafety, which is not ideal for stem cell research or clinical application. Second, animal origin-free culture system support consistent and regulatory compliant cell production. And finally, StemFit provides AOF culture system with highly efficient cloning performance, and significant cost reduction. So that's all. Thank you for your attention.